going to talk about Lab 6 because Lab 5 was your practical, so we're skipping to Lab 6. If you go to the homepage, there is just one document to read, but the format changed it a little bit. James is going to upload a PowerPoint and an introduction video to that PowerPoint so that you guys can walk through things. But we're also going to present so you guys can get it in multiple ways. This rapid differential median rapid identification document is going to pretty much have all or a vast majority of the information that you guys are going to need today, and I will tell you it is a lot, but we're going to try to go through it step by step so you guys can learn that tonight. Identification of unknown bacteria. This is the simulation like normal, and then there's quiz 5 at the end. So we're going to jump over here into the Jamboard. I can make this full screen here just a second. Great. Feel free to ask any questions as they come up. I should be able to see those. But like I said, there's going to be a lot of information tonight that's all going to be very similar. So really concentrating on how you will best learn how to differentiate between 12 different tests that we're going to talk about today. Each of these 12 tests, we're going to answer either these 3 questions if they're about the differential media, or these 3 questions if they're about biochemical tests. But overall, the overarching theme is identification of unknown bacteria. So this is the goal of all these tests. Each of these tests will not tell you what the bacteria is, but each of them will provide information about the bacteria that you should be able to, if combined with all these other tests, decide what kind of unknown bacteria that you have. And this is the exact same thing that you guys are going to be doing in the simulation. So figuring out the best way for you to simplify, really understand what the answers to these 3 questions will be the best way to study this type of material. So we're just going to jump right in. We're going to go over these 12 tests. This is just a print screen from one of James's slides. So I did some copying and pasting. So if you're following along in the PowerPoint, it may not be in the same order, but you should be able to see these slides and follow along. And so these are the 12 different tests that you guys know by the time that we are done tonight, and make sure you know these this week so that you can take this quiz. And we're going to be using these tests for the rest of the semester. So the first test we're going to talk about is the Catalase Test. The Catalase Test is testing for the catalase enzyme. Enzyme. I can't spell. So the catalase enzyme is what breaks down H2O2, which is hydrogen peroxide, to H2O and oxygen. So H2O2, hydrogen peroxide, this is the same stuff that you guys probably have in your medicine cabinet at home. It's also the same component that you would use when bleaching your hair, or you can also use it if you're getting into doing metal digests to identify heavy metals in samples. You can use hydrogen peroxide to digest metals at various concentrations. The concentration in your cabinet at home is pretty low concentration. I think it's at 3% usually. So hydrogen peroxide is a byproduct of cellular respiration, and it's one of those toxic reactive oxygen species that are produced. When it's produced, some bacteria that have catalase, are able to break it down into water, which is a liquid, and oxygen, which is a gas. So it starts out as a liquid, and it turns into a liquid and a gas. When you change something from a liquid to a liquid and a gas, you basically get bubble formation. If the catalase enzyme is not present, H2O2 doesn't actually do anything and just sticks around, and you still have hydrogen peroxide, which is pretty toxic to a lot of cells. So cells want to be able to get rid of H2O2 or hydrogen peroxide, and this is one of the ways they do it, through this catalase enzyme. So this test would be positive for the catalase enzyme if you see bubbles. No bubbles, it would be negative for the catalase enzyme. And what does this tell you about the microbe? It just tells you whether or not it has the catalase enzyme. These little paragraphs up here, I think that these I copied from the simulation. So you're going to be talking about this in the simulation, but I'll also put up here as we go through these some of the slides from that a series of slides that James is presenting for you guys. So these are the things you have to remember about the catalase test. It's looking for that catalase enzyme. This is what it is if it's positive or negative, and what does that tell us about the bacteria. Number 2, the oxidase test. So oxidase tests, we're looking for something called cytochrome C oxidase. And cytochrome C oxidase is a enzyme within the electron transport chain, ETC for short. Bacteria that utilize oxygen as an electron acceptor in this electron transport chain have this cytochrome C oxidase enzyme. How does this test work? The oxidase test, you take a sample of your bacteria and you basically put it on a test strip. This test strip has a reagent on it that will change colors if it is oxidized. I may not be spelling that right. So if this oxidase exists, it should change the colors on the test strip if it's oxidized. If it comes out a pink color, that means it's negative. And if it comes out a blue color, that would mean positive. And what does this tell us about our microbes? This would tell us if potentially they have this enzyme. I'm just going to write CCO for short because I don't want to write it out again. Cytochrome C oxidase. And if a bacteria has cytochrome C oxidase, because it's part of this electron transport chain, most often any bacteria that has this enzyme is going to be an aerobic bacteria. This is not a definitive, but it is definitely indicative that you could potentially have an aerobic bacteria or a bacteria that is able to utilize oxygen on your hands. So if you found something in your beer making that is in an anaerobic situation, but you tested it and it has the cytochrome C oxidase, you might suspect that it is a facultative anaerobic bacteria because it can survive breathing oxygen, or it could survive in an anaerobic conditions. So that's what an oxidase or the oxidase test is testing for. So definitely for each of these tests, know what a positive and a negative result looks like. And there's going to be lots of them. There's 12 total. So the Indole test, we're going to actually see this in 2 different locations in this PowerPoint. One of them is going to be using this DMACA, and then the other one is using DMABA. but we're not quite there yet. Just as a side note. So the chemical tested in the Indole test is we're looking for the use of tryptophan by the bacteria. So this is one of our amino acids. And if the bacteria is able to convert this to indole. So some bacteria can utilize some carbon sources, other bacteria utilize other carbon sources or nitrogen sources, and we can classify bacteria based on what they can use. So if the bacteria can use tryptophan, it will produce indole, and then we can classify it as a bacteria that can use tryptophan. How does this work? You would essentially, in this particular version of the test with a test strip, the test strip has this DMACA on it. This is the reagent. It's p-dimethylamine cinnamaldehyde. You probably don't have to remember that, except for if you were asked, you know, what it is. It's abbreviated as DMACA. So you can probably get that from this very long word here. And then the DMACA, if it is positive for indole, it will turn blue. And then if it stays pink or a light color, it is negative for indole. What does this tell us about the microbe? It tells us if the microbe can utilize tryptophan as a nutrient, as a food source. The second version of this that we're going to talk about a little bit later in one of our augers utilizes a test tube. In the test tube, in our test tube, we have a culture of bacteria. To this test tube, we add the DMABA reagent. This reagent, if it is pink, if it turns pink or red, it's like a reddish color, then it is positive for indole. If it is clear, then it is negative for indole. So there's actually two different, you can test it two different ways, and there's two different sets of color results as a result of whether or not indole is present. So that is the Indole test. The next question, or the next ones, we're on number 4 here, is nitrite reduction. So like some bacteria can use oxygen to produce ATP, some bacteria can reduce nitrogen to nitrite, to NO2 or nitrite, to produce energy. So the chemical that we're testing for in this particular situation, we're testing for the degradation of nitrate to, or the reduction of NO3 to NO2, and whether or not this is occurring by the bacteria. So when we're doing this, this is actually a 2-step process. The first step, so let's put step 1 here, we would add nitrate A and B. So these are 2 different forms of nitrate. And if when you add these 2 forms of nitrate, a red color comes up, it means it is positive for nitrate reduction. That means whatever this bacteria is, if you add these and it turns red, this bacteria is utilizing nitrate as an electron acceptor and producing energy. However, if it is colorless, I believe, or no color, it does not necessarily mean you don't have nitrate reduction. That's why you need step 2. So step 2, if it's colorless, it either means that this process is not occurring at all, or that this process is occurring so efficiently that this is actually continuing on down this line and producing nitrogen gas, and that gas is essentially floating away and you're not detecting this middle step. So in order to test to see if your nitrate reducer is just overly efficient, you would have this step 2 where you're adding in nitrate C to double check to see if this process is occurring. And this is where this might get a little bit confusing. In step 2, if it remains colorless, the reagent that you're adding in step 2 forces this reaction to occur if it is occurring. So if it remains colorless, it means that there is no NO3 in the system that can be converted to NO2, because this zinc powder forces any NO3 left in the tube to become NO2. So if it's colorless, it means that there's no NO3 left, and you have been so efficient at this process that you've produced N2 gas, and you're not detecting this middle step. So this is positive for nitrate reduction. On the other hand, if it turns red, because it was initially colorless, if it turns red, it means that NO3 was still there, and you've just forced it to be converted to NO2, and the bacteria are not doing that. So this is negative for NO3 reduction. So if, let's separate these two steps here. In the first step, it turns red. You don't have to continue because you already know that your microbe is performing nitrate reduction. If in step 1 you have a colorless solution, you do need to go to step 2 to double check if it is occurring, or if it is just occurring so efficiently that you're not able to detect it in step 1. So what does this tell us about the microbe? This tells us if the microbe can utilize a nitrate as an electron acceptor. The thing to note about this, if a bacteria is using nitrates as an electron acceptor, it's usually in an anaerobic situation, because oxygen is such a better electron acceptor that most bacteria will automatically choose to use oxygen. Some bacteria have evolved to use nitrate because they're not as competitive in an oxygen environment. This is really important, nitrate reduction, in wastewater management, in different things like soil microbiology, ponds, water management in general, to see if you're having these processes occur. So I'll write wastewater. Is really where this process is very important to be able to test for. So that was number 4. Next one. So now we're going to switch gears a little bit. Each of these previous 4 are purely biochemical tests. So we had the Catalase Test looking for the breakdown of hydrogen peroxide. We had the Oxidase Test looking for the oxidation by this electron chain acceptor enzyme. We had the Indole Test where you're looking for the breakdown of tryptophan because it's being eaten. And you have Nitrite Reduction, the breakdown of nitrate to eventually N2 gas as an electron acceptor. These next, so that was 4. So the next 8 that we're going to talk about are different types of culture media. So I know we've gone through this previously. We have our nutritive media. I just want to note that this is a general purpose media. So general purpose media is the phrase I've used before. They're essentially the same thing. Again, your enrichment, you're providing, so you're adding in, selective, you're inhibiting, and differential media. So all of these, I believe, are no, not all of them, actually. Most of these are differential media where you're distinguishing one group from another. So those are kind of those keywords to look for. This one's like most bacteria or all bacteria. So the first one we're going to talk about is the blood agar plate. Composition of the media. The important component of this media is sheep's blood. So there's actually blood cells in this media. That's why it gets that red color. How does this work? So this particular media, because it has the sheep's blood, it's an enrichment media. Because you're adding in an ingredient to see if that helps certain types of bacteria grow. It's also a differential media. So differential means that we can tell the difference between different groups of bacteria based on how they grow on this plate. So how does this work? The bacteria can grow on the plate, and you can differentiate based on hemolysis pattern. So hemolysis, you can imagine you have a little blood cell here floating around in the media. Depression in it. I can't draw a blood cell apparently. But some bacteria can introduce things that are called porins. P-O-R-I-N-S. Into red blood cells and basically cause them to lyse or break open. And they can extract the iron within the cell. So hemolysis is the breakdown of those blood cells that are in the agar. And there's 3 different patterns of hemolysis that you can differentiate. So I'm just going to zoom in here, but this is on that PowerPoint. So A, B, and C are 3 different types of bacteria on this plate. A shows what's known as beta hemolysis. So beta hemolysis is complete lysis. If you held this up to a light, you would be able to see the light through the plate because you've completely broken down those blood cells. B, growing here, demonstrates alpha hemolysis. So alpha hemolysis is partial hemolysis. You can see there's a little bit of that glowing of light coming through the plate here, but mostly it's pretty cloudy and you wouldn't be able to see through it if you held it up to a light. So on here it shows that there's like slight ring around it, but it's definitely not the clearing that you see with the beta hemolysis. And C is finally a bacteria that does not demonstrate hemolysis. So this bacteria just essentially is dark on the plate, and this is known as gamma hemolysis. So there's no hemolysis that occurred. So there's alpha, partial, beta, complete, and gamma, which is no hemolysis. And what does this tell us about our microbes? It tells us if they can utilize blood as a source of nutrients. One of the big bacteria that would be growing on a blood agar plate showing hemolysis patterns would be the group Staphylococcus. So Staphylococcus aureus is a bacteria that you can find on your skin. It's also the SA in the term MRSA. So this is an important hospital-acquired infection where Staphylococcus aureus, this SA part of this word, is methicillin resistant. So this is an antibiotic-resistant Staphylococcus aureus. And I believe a Staphylococcus aureus would show the beta hemolysis pattern. So that's just one type of bacteria that would be useful to grow on a blood agar plate. The next type of media that we're going to talk about is the MAC media, or the Mcconkey Auger, so MAC media. Mcconkey Auger contains bile salts and crystal violet. Bile salts and crystal violet are both gram-positive inhibitors, so this is a selective media because they're inhibiting the growth of gram-positives. Therefore, if you were asked a question, what does a gram-positive bacteria look like on a Mcconkey Auger plate, it would not look like anything because it wouldn't be growing. It would be a blank plate. Because of the bile salts and crystal violet, you should only get gram-negative growth on a Mcconkey Auger plate. And it also has the neutral red indicator that changes colors depending upon the pH of the plates. So how does this work? This particular plate tests for lactose fermentation. So some gram-negatives can ferment lactose. So if lactose, which is a sugar, is fermented, then I think the plate becomes acidic, and then the neutral dye will change colors. So if there is no fermentation, it is colorless, is negative, and if there's fermentation that occurs, red is positive. And actually, I think I got that wrong. I think it's alkaline. I have to double check that. So what does this tell us about our bacteria? This would tell us, first of all, that it's gram-negative if it's growing on this plate, and that it is whether or not it is a lactose fermenter. Bacteria, an important group of bacteria that is gram-negative and lactose fermenters are the entero, E-N-T-E-R-O bacteria. Enterobacteria are gram-negative lactose fermenting rods, and these are important gut bacteria, and they cause a lot of food poisoning that you'd see. So things like E. coli and Salmonella would all be part of the enterobacteria group. So that's an important group of bacteria that you might be testing for with a Mcconkey Auger plate. The next one we're going to talk about, so that was our number, what number are we on? We were 4, blood agar was 5, Mcconkey is 6. We're on to number 7 here, of 7 of 12. The mannitol salt agar plate. This one, it's pretty straightforward based on the name. It has a mannitol as a sugar. It has salt, and it also has phenol red is the indicator dye. So we add mannitol because we're adding mannitol as a sugar source. Sorry, because we're adding salt. Salt is not a nutrient. Salt actually affects the osmotic pressure of the cells, as we learned about previously. So any media that has salt in it would be a selective media. And because it has this phenol red indicator, this would also be a differential media. How does this work? If a bacteria is able to grow on the salt plate, it tells us that it's halotolerant. And then if the bacteria is able to use a mannitol as a food source, or as a carbon source, basically, if it is able to ferment mannitol, it will be yellow. Yellow is positive for fermentation. And if it stays red, because the original plate itself is red, it's an indication of negative, that the bacteria is not able to utilize mannitol as a food source. So again, in this example of the slide I took from James's PowerPoint, you're looking at different types of Staphylococcus, which we were just talking about, for potential mannitol fermentation as well. So they ferment potentially lactose and mannitol. Actually, I don't think they ferment lactose. That we were talking about Staphylococcus aureus with the blood agar plate. So with your, what does this tell us about these bacteria is that, first of all, if they're growing on here, they're halotolerant at least. They could be halophilic, but they can survive the 7.5% salt. And it tells us if they can use mannitol as a food source. So again, each of these tests does not tell us definitively that what our unknown bacteria is, but it adds another piece of data that we can use in combination with other tests to really identify and narrow down what we have. So our next one, number 8 here, is our PEA or P-agar. It is phenylethyl alcohol agar. So this is actually our only non-differential media. This is actually just a selective media. And the composition of this media is that it has this phenylethyl alcohol. Phenylethyl alcohol, because it's an alcohol, will disrupt membranes, lipid membranes. So think in your gram stain, you use the grams alcohol, and it washes that outer membrane off of your gram-negative bacteria. So our little gram-negative bacteria have that inner thin peptidoglycan layer, and then they have that outer lipid membrane. So this is a gram-negative. It's a really bad picture of it. So this particular agar, because it has this phenylethyl alcohol and it'll disrupt lipid membranes, it'll actually disrupt this outer membrane on gram-negatives and cause them not to grow. So it is actually selective for gram-positive. So if there's growth, it is positive, and it's indicative that it's gram-positive. And if there's no growth, it'd be negative. So these results over here, you see the A and C are growing. B is growing a little bit, so it's not a definitive that it's not going to let any gram-negatives grow, but it's going to severely reduce their growth on these plates and select for gram-positive growth. So that's our P-agar. Next one, so let's see, number around 9. So number 9 here, bile escalin. a bile escalin, you can notice here that our previous plates we were talking about were actually microplates. So these circle plates that you've been using in simulations as well. We're also going to talk about nutrient media that we can grow in tubes. And sometimes the tubes are helpful in identifying other components of bacteria as they're growing. So bile escalin contains escalin, contains bile salts, escalin, and ferric ammonium citrate. Bile salts, as you remember from our Mcconkey Auger, I believe. Bile salts in crystal violet and Mcconkey Auger inhibit gram-positive growth. So this is selective because it inhibits for gram-negative bacteria. And it is also differential because we can identify whether or not bacteria can utilize this escalin food source. How does it work? If the bacteria is able to grow on these slants, so they pour the agar in the tube and then you can plate the bacteria along these slants. If it is able to grow on the slants, if it converts escalin, if it's able to utilize escalin as a food source and convert it into glucose and esculatin, this is SCULIN, converted to D-glucose, which is a simple sugar, and esculatin. And then it's continuing able to utilize the sugars. If it can do this, the esculatin byproduct will react with the ferric ammonium citrate and produce a black precipitate. So this has iron in it, and iron reacting with different things will turn black in a media. So if it is black, it is positive for this esculatin production, and it means that the bacteria can use escalin. If it stays brown, it is negative. So you can see in this picture here, it's like a reddish brown. I would call that brown. And what does this tell you about the bacteria? First of all, it's probably, if it is growing in there, gram-negative, as well as whether or not it can use this escalin as a food source. And you might think, why is that important? It is apparently one of the only ways that we're really able to have a simple test to differentiate biochemically between Enterococcus and Streptococcus. So Enterococcus and Streptococcus back in the day were the same family or the same genus of bacteria, and then they continued to split them based on certain differences. So this is one test that you would be able to perform to tell which category they belong to, the Enterococcus or the Streptococcus. Great. Number 10. So our last 3 here, 10, 11, and 12. This one's pretty fun. This one is SIM Auger, which stands for Sulfur Indole Motility Auger. So it's actually 3 tests in one, which is why you see the 3 slides over here. And the first slide, or the first test that we're going to talk about is sulfur or sulfate reduction. So let's zoom in here. So sulfur reduction, some bacteria can utilize sulfur from cysteine, which is one of your amino acids, or can reduce any sulfur that you add directly. And again, this is similar to your nitrate reduction. You can have a sulfate reduction in bacteria. So the reduction of sulfur produces a gas, H2S, hydrogen sulfide. And H2S will interact with ferrous sulfate, which is, again, that iron substance in the media that will turn a black color. So let's log out here a bit. So composition of the media. It has ferrous sulfate in it. And it has some form of citrate or other type of sulfur. So that would be supplied through the ferrous sulfate. And it is testing, so this is, let's just put this as our first test here. It is testing for sulfur reduction to H2S, which is a gas that reacts with the ferrous sulfate to form a black precipitate. So black would be positive, clear or colorless would be negative for sulfur reduction. Our second test, so our second test is testing for indole. We actually already talked about this. So this composition of the media contains tryptophan. Indole, or that DMABA, is added to the tops of these tubes. Let's zoom in on this one. DMABA, which is known as Kovacs reagent. So if you're asked about Kovacs reagent, this is an indole test to test to see if indole is present for the breakdown of that tryptophan to indole. So if you add it and it turns red, that means it's positive. If it remains clear or kind of clearish, it would be negative for indole production. So red is positive, colorless is negative, and that's after we add that DMABA, which is Kovacs reagent. The third test that we're looking at here is for motility. There's actually nothing that we add to the media that would test for motility, but it would tell us whether or not the bacteria. So the first one, if it can reduce sulfur. Second one, if it can utilize tryptophan. And the third one would be whether or not it's motile. And as we were just learning in lab before, I believe, motility is through those flagella that are present on our bacteria. So if we were, when you plate, and this is why the tube format is helpful, when you pour these tubes, you can pour them directly and you basically stab a needle that is inoculated with your bacteria into the media. If the bacteria stays and grows along that stab line, it means that the bacteria can't swim. If it goes out into the media and becomes kind of cloudy, it means the bacteria is able to move and disperses into the media. So this would be positive for motility. This would be negative for motility. So for this media, it is differential because it is, let's write this down first, differential. It's a differential media. It's not really selective or anything. It does have a tryptophan, so you could argue that it's an enriched media, and that helps those that can utilize tryptophan to grow. But overall, we're testing for these three different things: whether or not it produces that black precipitate, whether or not it turns red when you add Kovacs reagent, and whether or not the bacteria grow along that stab line or if they move out into the media. So if we take a look at some of our other pictures up here, we can see in this culture here, we have the stab line of bacteria. They did not grow out. So that would be a non-motile bacteria. It is also negative for sulfur reduction because there's no black precipitate. And assuming they've already added Kovacs reagent, it would also be negative for indole production. This one over here, you have positive sulfur reduction because you see that black precipitate. You also can see where they stabbed it and that black goes out into the media. This means that it was motile or positive for motility. And if they've added Kovacs reagent, it is negative for indole production. Down here, it's kind of a bad picture. This one, it's positive for motility because it's swam out into the surrounding colony. It's black, so it's positive sulfur reduction, and it's clear at the top, so it's negative for tryptophan. This one, positive for tryptophan because it's that red line at the top. You can see the stab line here is actually the bacteria growing along it, so it's negative for motility. It's also negative for sulfur reduction. So there's 3 different things to assess whenever you're reading something in Sim Auger. So it's pretty useful. It's a pretty useful auger. I think that was 10. Last 2 are pretty simple overall. Number 11 is the Urea Agar. You're looking for the presence of the urease enzyme. And if a bacteria has the urease enzyme, it can convert urea, U-R-E-A, to ammonia. And this causes an alkaline pH to form. So it changes the pH, C-H-A-N-G-E-S, the pH. And when you have this pH change, you get the color change because of the phenol red indicator. So this has, we're testing for the urease enzyme. The media itself has urea, peptones, glucose, phenol red, indicator dye. What does this tell you about the bacteria? So if it turns pink, it is positive. If it stays yellow, it's negative for ammonia production or the breakdown of urea. What does this tell us about the microbe? Whether or not they have this, the urease enzyme. Okay, and last but not least, our Uncitrate Auger. So Uncitrate Auger, I don't think we can talk about that in here. So this one would be a differential. It would also potentially be arguably an enrichment auger because you're providing that urea enzyme or that urea food source. Last but not least, Uncitrate Auger, very similar to your Urea Agar, or your Urea Agar, in that the Uncitrate Auger is testing for the citrase enzyme. Which is able, if they're able to break down citrase, use it as a source of carbon, you will produce ammonia again. And this will cause an alkaline pH. So a blue color in this case is green to blue. Yeah, a blue color would be positive, and a green color, I can't spell green. would be negative. So apologies to anybody who's colorblind, but these are the colors that come out of a Uncitrate Auger test, blue or green. So the media itself has citrate as the source of carbon. And it has this bromothymol blue indicator dye. Those are two of the things that are in this auger, and overall it's answering the question of whether or not the bacteria have this citrase enzyme or this pathway to breaking down citrate as a carbon source. So this is also a differential media. And you could argue a selective media as well. Well, not a selective, enrichment media. You might be able to argue it's selective, but you'd have to give me a good reason why, and I'm not going to tell you what that reason is. So those were the 12 tests that we started with at the beginning. This is how it looks, some of the tests that you're going to be doing in the simulation itself. So you guys are going to be going over your differential staining, which we haven't talked about. But remember, we had those 6 different stains that we used. Differential media and our biochemical tests. So we just learned 12 different differential media and biochemical tests that you guys will use some of within the simulation. So overall, the point of running all of these tests is to be able to utilize a table similar to this with known properties of different bacteria and be able to narrow down utilizing these tests. For example, if you ran an oxidase test and it came back negative, you would know it's not Bacillus subtletes, so you could cross that one off. To narrow down overall what your bacteria might be. So this is overall, if you were going to identify an unknown bacteria, you would want to utilize some sort of a table or some sort of a key where you had known properties of different bacteria and be able to narrow down using these test results. That's really all I wanted to go over with you guys. Definitely take a look at James's PowerPoint. He presents this information as well, so there might be some different ways of seeing the information that might help you. So definitely listen through that at least once and really come up with that way that you guys are going to be able to remember what positive looks like, what negative looks like, what is in the media, and what is it telling us about these bacteria. So I know it's a lot of information, but you guys are smart, and I have full confidence that you guys will be able to start working through this information. We're going to be using this throughout the next half of